attendance and for inviting me. And then it is something that really dear to my heart because you're talking about internship, especially summer internship. I had my experience of a summer intern, but let me start with something else. Actually, I prepared something to say because I have a tendency to drift along in all directions and things you may not want to hear. Okay, right. So I will comment on the topic I was assigned, but let me say something else. So first, I start with my own experience in summer intern programs, as I mentioned earlier. Then I say a few words about science and technology, and followed by STEM education. Okay. And then actually my first scientific investigation happened when I was in primary 2. Of course, not summer, and then I use anything possible to do experiments. Some of you may know that I grew up in a farm, or near a farm, and so I had much chance to see the nature, growing silkworm, and then watching them turn into moth and then so on. It was rather simple because you get the silkworm from the vendors and then you pick the mulberry leaves and feed them and watch them and so on. But catching tadpoles and watch them to metamorph into a frog, imagine. I couldn't imagine either because it never happened. Didn't realize that when you catch a tadpole, you have to feed them. What do they eat? Little mosquito larvae. We don't have mosquito larvae. Well, unless you catch a lot of them yourself. And then you don't eat anything else, just that. So we never see the tadpole turn into a frog. Okay? And then I also use my sister's toy kitchen utensils to cook things. Chemicals. And I learned that when you burn sugar, you can turn them into caramel, right? So I did that, of course, ruined my sister's utensils. Anyway, and then I did a little bit more sort of biological experiments. The teacher said different beans would germinate at a different speed, okay? And then we're supposed to record how they perform. And then so I watched these beans and the broad beans, red beans, green beans, whatever. And then for 2 weeks. And it was great fun. That was primary 2. Okay? Then my first summer internship actually came, not primary 2, but the second year of university, or second year of college. That was in 1969. I had a chance to work in a molecular biology lab, the lab of Professor K.K. Mark. And then I didn't just wash dishes here, as many of you imagine that you work in a lab, you wash dishes. In those days, there weren't too many disposables, so you really have to wash everything. Anyway, so we didn't get to wash dishes. Well, of course, not disappointed. We were asked to produce a so-called one-step curve. I don't know how many of you heard about that. Yeah, any of you? It turns out that the organism we're studying was a bacteriophage, or a virus that infects bacterium. Okay? So we in fact use, of course, an inoculation of a viral sample into a bacterial culture and watch how they burst. Okay, the virus gains the bacteria and they will burst. So from one infected cell or one virus, you get hundreds, if not thousands. So one step. If you do it properly, of course, you have to do in a culture with many viruses and many bacteria, bacterial cells. So you really have to count, right? There are ways to count, but I'm not going to get into that. So if you do that properly, you sample every about 2 minutes or 10 minutes, and then you watch the number of virus in the culture burst. Okay, so one step grow. And my curve was beautiful. So the best in the group. So I said, the happiness of seeing such results at a bench was really difficult to describe. And I guess that's why I chose a research career afterwards. That was my one and only summer intern experience. Now, you work in a real research lab or any research lab setting, laboratory setting, is different from working in a science class. I mean, in a science class, you have all the things described clearly. You're supposed to see this, you're supposed to see that. But in a real laboratory or research lab, you cannot anticipate what you're going to see. Okay? There's no set answers to the experiment. So you must record all results, right? And then don't throw away any spurious data. Some may think that you're supposed to see a one-step curve, right? Then what happens? You see a slope like this, and then you say, oh, that's bad. You throw away the unwanted data to produce the one-step curve. Then that's actually bad. The teacher may trick you. They may have different strains of viruses in there, then they may come out to be a slope instead of one step, okay? So therefore, really be honest to your experiment, to be honest with your data. That's a very important thing. And then do not just try to fit a strict line or a sort of a prescribed curve, and then just honest with your finding. In fact, a lot of very key experiments are based on small, or key discoveries are based on small discoveries, the things people did not throw away. And then they look at it, and then you have a huge discovery. All right? So my second point is about science and technology. The work of science is to discover the law of nature, how things happen, etc., etc. I know how you do that. So the scientist's job is to discover. Whereas technology or scientific technologies is just to apply scientific principles to build or invent new ways to do things to benefit all of us. Therefore, engineers are inventors, right? So scientists are discoverers. Then sometimes scientific investigation and technology development, they go hand in hand. I should say often they go hand in hand. So you have new technology, you can do some new science investigation, and new science investigation, you have new technology. And then, but then sometimes in Chinese, the two things got mixed up. You cannot tell what's science, what's technology. Okay? For example, we have the 4 big inventions in China, right? That's all technology. It's not science. And then Joseph Needham didn't get it right either. So, well, of course he did. And then, we have a university here in Hong Kong. It's called University of Science and Technology, but in Chinese, 香港科技大学. 科技 became one description. Anyway, so I hope that you're doing science in the future. Of course, you're learning different tricks, but you also learn what is science. And it's very important. When other people, if you happen to work in a lab, of course, I know that your summer internship work on many different things. So you work in a lab, so just don't tell people, I learned such and such technique, okay? Nobody care what technique you use. People would care what scientific problem you're working on, all right? So, and besides, techniques, they change from time to time. And then this year you have this popular technique, and then next year they have new ones. And then a lot of techniques, you have so-called kits to perform, and then you don't need to think what's inside. Even the gene editing, you can buy kits, and then you can edit genes yourself, at home even, all right? So while talking about science and technology, I must say a word about innovation, okay? We are talking about scientific innovation. Hong Kong has a Innovation and Science Technology Bureau, so therefore in Hong Kong, innovation goes before technology. But I must say that we must have science and technology go before innovation, then you have new discoveries. Now that's a little bit of my own belief, and then therefore you really have to know your science and technology before you come to innovation. But then someone actually tried to interpret or to describe what innovation is about. So this professor used to be in Toronto, later on they went to UNC, and is a she said, what's the difference between innovation and invention? So invention is the creation of a new concept. Innovation is reducing that concept to practice and making it into a commercial success. Okay, you can check out this Marianne Feldman. That's what she said. And then the next thing she said about innovation versus creativity. Now, in Hong Kong, I think we all know that what is creativity? It's just coming up with ideas. Okay? And then the innovation is a step further. It's bringing ideas to life. Okay? And then she also talked about innovation and science. I love this one. Science is the conversion of money into knowledge. You spend money to do science. And then now you know what the other side, innovation. Innovation is the conversion of knowledge into money. Right? But therefore, I think I don't blame the Hong Kong government by putting innovation in front of science and technology. All right? So my third comment is about the so-called STEM, S-T-E-M. You all know what the STEM stands for, S-T-E-M, right? But I talk about this many, many times, but at this occasion, because recently in Hong Kong, in particular, they talk about STEAM, S-T-E-A-M. They thought S-T-E-M wasn't enough, and then we have to add arts in there. Okay, because S-T-E-M is all about science and math and so on. You left out arts. And then later on, they put R somewhere in there, became stream. They said reading is also important. I said, what's not important in high school? The original idea of STEM education came from the US when Obama was seeing the students in US, they are not taking science seriously. Okay, so US have been lacking in a lot of scientific investigations, and then they have to thank a lot of foreign students. They are still relying on a lot of foreign students. But anyway, so they want their own product. They want especially by turning all the scientific knowledge into innovation. So how do you do that? You must put things together. You don't study biology or just study mathematics. If you can put mathematics and biology together, you create something new. Okay, so-called the interdisciplinary, cross-disciplinary kind of investigation. That's how STEM idea came out. It's while you're studying biology, you think about how can you use math to improve? How can you use computer or AI technology to help improving the research? And when you're doing AI, you say, are there any questions in biology I can help? So you're still studying AI seriously or studying biology seriously. Or for that, you study chemistry, physics the same way. You are doing one study very deep, seriously, you have your mind on other subjects. It's not like you just go into a lab and play with a 3D printer or play with some robots, and then you finish your STEM education. All right, so STEM, whatever STEAM or STREAM, are just philosophies in education. All right, so just keep that in mind, especially when you see the younger generation in Hong Kong. They say, I study STEM. Yeah, right, you play with something in the lab. Anyway, I should not say I get sidetracked too much. I must remember our original purpose here. Therefore, I must congratulate the Hong Kong Life Sciences Society for initiating this summer internship program, because really this is a very effective way of letting you have a taste of scientific research or work in the area of science and technology. In this case, as probably life sciences, I must say that. Okay, so in the remaining time, I have to answer the questions. Where are we in basic life sciences research? And then the Undersecretary already mentioned that the Hong Kong government had invest quite a lot in the biomedicine or biomedical research, biotechnology rather. And then in fact, we all say that this century belonged to biomedical research, biomedical sciences. Although some would add AI at this moment because AI became very popular. But as a whole, the previous century is more about physical science, engineering, and so on, right? So this century is all surrounding the biomedical or life sciences. So, and then we have really learned a lot in the past 20, 30 years, especially since the completion of the Human Genome Project. So note, this is the Human Genome Project. The project has finished. The human genome has not finished. Okay? So in fact, there's not a single human genome has been sequenced, documented from one end to the other. We have 23 chromosomes. No, not one single human being has been fully sequenced. Okay? So the project is finished. It's just the beginning of using the knowledge that we have to do as an investigation, as a starting point to have more investigations. There's still a bit of unknowns. Yeah, so not just a sequence of genes, I must say that the genes do not live alone in a cell. Okay, first we have two pairs of chromosomes, right? So one from one parent, the other from the other parent. So we have roughly 20,000 genes, therefore 40,000 genes interacting with each other. And then we have some simple idea of how they work each by themselves. But how they work together, we're only beginning to understand. So therefore, when we talk about the human genome, people quickly switch into, oh, we are now into proteomics. Study of proteins. Of course, a lot of gene products are proteins, so we study how proteins interact and so on. So I don't want to bore you with all the details, but then there's really a lot of things in front of us to explore. Life sciences really is going to flourish in front of your eyes. And then the second is from basic research. So you use basic to scientific, to translational research, okay? I use myself as an example. We started to clone the gene called cystic fibrosis, causing the disease cystic fibrosis. After some years, eventually in 1989, we managed to clone the gene. That was before the Human Genome Project. So a lot of work, a lot of firsts, and so on. But it took actually some time for us to realize what was the function of the gene. We had a sequence, but we had no idea what the function was. But of course, at the end, it was quite clear that the protein that's encoded by this gene that we cloned that causes cystic fibrosis is a chloride channel. So it's a small chloride channel that works in the apical membrane of a secretory epithelial cell. Okay, a mouthful. But anyway, so that regulates the secretion of exocrine organs. Anyway, so because of a small protein defect, okay, that control or regulates the flow of chloride ion in and out of a cell, okay, or in and out of your body, and that could cause a big problem. The disease is rather complicated. Patients with cystic fibrosis will suffer from lung infection, intestinal indigestion, and many other things. So very complicated. But because of the cloning of the gene, people are able to hone in to study what was wrong with this in a very detailed way, and how can one actually find ways to cure the patients. I said, wait a minute, a genetic disease, how can you cure a genetic disease? Use gene therapy, right? So immediately after cloning of the gene, people talk about gene therapy. So 5 years we have gene therapy, but didn't realize that the gene regulation is rather complicated matter. It's not that easy. So gene therapy was very nice, the genetic defect gene therapy. And now people talk about gene editing. But at the end, you know what? After almost 30 years, now there are small molecules that scientists identified. These molecules can correct the basic problem of these proteins, these channel things. Okay, the channel just won't let the chloride ion go in and out of the cell. But then with these small molecules, the patients, after treating with these small molecules, they behave like normal individuals. So that's almost like cure, right? So that really, to me, is kind of a miracle. But our contribution was the cloning of the gene. Eventually, we went to study of the different mutations. We classified the mutation. We studied gene-gene interaction and all these scientific things. But then other scientists study the channel properties, and yet other scientists look for the small molecules. So all these steps, it took almost 30 years, right? But a lot of people, one time I joke, there were probably more scientists working on this CFTR molecule than patients. Of course, now with the treatment, and all the patients have cure. Okay, so in other words, it's not easy to go from a basic discovery into or translate into a product. So then what are we lacking? So in Hong Kong, that we have lots of people working on basic research. The Hong Kong government is promoting innovation technology in life sciences and hoping that our universities will help to develop a new ecosystem to create jobs and wealth for the region. Or, of course, now everyone talk about in collaboration with the Greater Bay Area. So obviously, this is a very ambitious goal, from university research all the way to translation into products, into economy. So I sometimes say it may be an impossible dream, but it's important because, like I said earlier, life sciences or biomedicine is the really science of this century. You have many more years to come. So it's not too late to start, right? So we do have very strong basic research in biomedical area in Hong Kong. And we do have a lot of other good and supporting mechanism, like the legal system, the investment environment, and so on. So, but then I always say that Hong Kong has very good upstream the research, and in this case we're talking about life sciences, but very few mid and downstream, and then very little experience. So, but I'm really glad to see the situation. The situation is changing in recent years with a huge investment from the Hong Kong government. I heard that the number of startups is increasing, and then there's also a lot of investments coming in Hong Kong. Even multinationals are coming to Hong Kong. So these are all very good news. But then this is very different from saying that every discovery or invention should go from upstream to downstream in a single laboratory. So that's not too many Dennis Low in the world. Okay, he was lucky. Then is because his little, not shouldn't say little, his invention got a recruitment company to support him, right? So therefore, essentially, it's a very simple technology. So it's an exception. So not everyone can be like that, okay? Of course, we are always looking for exceptions. So you could be one of the exceptions too. But then in general, you want to develop a so-called ecosystem, and you must have a lot of different things. Besides the supporting environment, as I said, the legal system, all the investment and all that. So you have very good upstream research, very good the so-called translational midstream activities, and also you have manufacturing companies on the other end or the downstream, okay? But it doesn't have to be A, B, C, or 1, 2, 3 in such a step. As long as you have enough activities in the region, not necessarily just in Hong Kong, but in the Greater Bay Area, but sometimes when they say Greater Bay Area, we include Shanghai and the rest of China. Oh, why not the rest of the world, right? So that should not have any boundaries. So therefore, you just work with the best people that you can, right, in the region. So the last thing I want to say is that please don't expect our professors to get into business. There could be exceptions. Last, I think I should stop here. I thank HKLSS again for providing this Life Sciences Summer Internship Program. And I wish all of you a happy summer, and then the students have a rewarding experience, and most of all, you find your career in life sciences in the future. Thank you very much.